Hello, hello. Whew. Thanks, guys. That was a great turnout. Thank you so much for your patience. Want to welcome everybody to the Connect. And thank you so much for endorsing Hydro for hosting us. Always appreciate you guys, Kevin and team. Really appreciate you. Thank you, guys. A little round of applause for Kevin and team. Thank you. So we have a very special guest tonight, hailing all the way from Canada. Good old Hygrozyme is here for us. Legendary Hygrozyme. We're going to be focusing on some root health optimization today, which for me, running a deep water culture nursery, is near and dear to my heart. It's all about the roots, keeping those roots healthy, pathogen prevention, clean behaviors. That's what it's about. So without further ado, I'm going to go ahead and introduce our panelists up here. Please go ahead and start off.
Hi, everyone. Thanks so much for coming out tonight. We're really excited to be here. My name's Leanne, and this is Doug, and we are from Hygrozyme. And yeah, so tonight, like Sergio said, we're going to be talking a little bit about root health optimization, a bit about why the root zone is so important, some of the common challenges, some tips and tricks, and then finally some solutions with our products as well. So with that, I'm going to kick it off to Doug to introduce himself.
Yeah, so before we start, we thought I'd give you a little bit introduction about ourselves. So my name is Doug Chow, and I've been with Hygrozyme for about 3 years. I started my career in completely different industry. I was in the pharmaceutical industry for about 12 years, developing a drug for heart disease. And then about 10 years ago, I migrated to the traditional agriculture industry. I was there for about 6 years doing traditional agriculture, developing biopesticide product for outdoor crops. And the products you might see in the market, I was mainly dealing with natural essential oil type of product, and they're approved. We launched 2 products, and they're actually also here in the US, and I think I seen one of the product in Calvin's shop as well too, one of the products. And joined Hygrozyme about 3 years ago, and yeah, and that's my background. And, you know, going from traditional ag to this industry, just a natural progression.
So I'm Leanne Spolek. I'm the Sales and Operations Coordinator for Hygrozyme. I've been with the company about 3 years, and I actually started in a production facility. So I've been in the industry for almost 7 years now, and I've gotten the opportunity to work in a lot of different departments within the industry, from operations to admin to project planning to being right in the greenhouses and trimming. So I've been really fortunate to see all the different sides of this industry, and I'm just really passionate about helping growers achieve the full potential of their plants. So super excited to be here tonight. And just a little bit about SIPCO and Hygrozyme. So SIPCO is a company started in 1974 back in Canada. And we actually started out with a lot of industrial products, industrial cleaning products. And then about 20 years ago, our CEO, Sheila, saw a trend towards green innovation, sustainable products. And so Hygrozyme was born, and so it's been in the market for about 20 years, has a really strong reputation, and since then we've been able to launch 3 additional products into our line, so 4 in total, and all of those products are backed by our ISO 9001 quality certification, which basically just maintains the quality and the consistency of all of the products that we ship out. And I'm going to hand it back to Doug to talk about some root health.
Okay, so as Sergio mentioned, we're here talking about root health today, and I'm sure I don't have to tell you guys in the audience how important the root is to the plant, you know, in particular this crop, right? So, you know, it's the root, it's an anchor for your plants, helps your plants to absorb nutrients, water. So you want to maintain your root nice and healthy, but it's also important to recognize when you do have issue with your roots, what are some of those issues, and how can you prevent them from forming in the first place. So preventative measure is really critical, but recognize some of the root disease and symptoms and things like that is really, really critical as well too. So over the next few slides, Leanne and I will walk you through some of the common root disease that you may have seen or you may have experienced personally in your grow. But so if you have any questions, during our chats, talks, please raise your hand and ask a question. This is it's interactive, so we want you guys to ask questions
Exactly, guys. This is a little bit different from the prior connects where we save all the questions till the end. This is a lot of great information, so please feel free to raise your hand, interrupt. Let's get that engagement going throughout the panel because I think that's a little bit different than we've been used to. So scream out my name, let me know, interrupt. You're good to go.
Yeah.
Thank you.
Yeah, so the first one I'm going to talk about is Fusarium. So hopefully, maybe some of you have seen it, and maybe hopefully some of you have not. But I'm going to tell you what it is. Fusarium is it's a soil-borne pathogen that causes a root rot, crown rot, wilt, and it's not just common to cannabis. It's common to all sorts of plants and crops, right? And some of the symptoms that you may see is yellowing foliage, shortening of the plants. You also get damping of the cuttings, and if you actually cut the stem and the xylem, you see necrosis of the xylem as well too. And then, how the disease is translated is typical, just like any other things, through equipment, through water, and just it could be even from you going to a different facility and you may not know it, and you go to another room, from one room to another, facility to another. So cleaning, preventative measures is really, really critical. And probably one of the most common way of infected is through the mother plants. So the mother plants might have the pathogen inside the xylem, but there's no symptoms. So if you take the cutting from there, then all your cutting is going to get infected with that, right? So if you see something that's not right with your plant, get tested, find out what it is, isolate them, and then once you know what it is, then you can start having having some solution for it.
And Doug, let's talk about you mentioned a couple of vectors, right? So mechanical transmission with your shears, waterborne pathogens are very scary, right?
Yes.
That can happen with temperature change. That can happen with not cleaning your drip lines or your emitters throughout runs or even maybe in between runs. You can talk about batch tank maintenance, right?
Yep.
There's a lot of pathogenic pressure that can develop over time, whether it's in your emitters, in your batch tank, or even just temperature. Let's say you lose some temperature control in your room, and what happens during that 1 or 2 hour break when you lose that VPD and the temperature spikes? Pathogenic breeding ground. And does anybody know what the temperature is when Fusarium can be actually sporulating? Is it in the mid 70s? Is it in the 80s? Do we have any scientists in the room?
Anybody?
Anthony? 72? Is that a guess or are you for sure? As long as you stayed at 68, you would kill it. We're going to fact check that, but I like where you're going with this. Thank you.
Yeah, like Sergio said, like in order for a disease to develop, you need 3 factors, right? The pathogens, there's what's called a disease triangle, right? Your pathogen, your crops, and your environment. Those 3 things will dictate the disease.
So the next one up is something I'm sure probably everyone in this room has encountered at some point or other, and that's Pythium or root rot. So of course, some of the obvious signs and symptoms are going to be brown roots that kind of slide off, lack of feeder roots. You might start getting an odor around your roots as well. Your plants are going to be a little bit stunted, leads to lower yield. Your leaves might start to wilt as well. And like Sergio mentioned just a couple minutes ago, this is a water mold, so it will spread through water. So if you are running DWC or something like that and it affects one plant, it is going to spread. And it can be very, very difficult to get rid of root rot once you have it. You can try and cut out some of the affected roots as much as possible, and you might be able to get away with that for sure. But once it's fully set in and your leaves start wilting and it starts moving into that crown rot, you may end up actually having to pull that plant entirely, which is why preventative measures and keeping an eye on your overwatering and your drainage and products like Hygrozyme can really help to prevent this from happening in your grow so that you don't have to lose any plants.
Okay, so the next one I'm going to talk about is I'm sure a lot of you have seen it. It's hop latent viroid, or so-called dudding disease, right? And some of the symptoms you'll see is the plants, if it's infected, might look completely different, like from a different genetics, right? And the leaves are thinning, as well. And obviously, you're going to get much lower yield and terpene. And the problem with this particular disease or the virus is that it's really difficult to tell the symptoms. Like if your whole growth system is infected, you wouldn't be able to tell because they all look the same. It's only you're going to be able to tell when you actually have some healthy plants and some that's infected. You'll see a big differences like shown here. As Leanne clicked through some of these slides here. And can you click one more? And so one of the study, this is actually a slide from one of the professors up in Canada at Simon Fraser University. We work with him quite a bit. Dr. Zamir Panja, he's a well-known plant pathologist in this area. So what he did was he actually took a healthy plant, inoculated the virus at one of the leaf, and then there's a progression of the virus over time. And you can see that 2 weeks is at the root. And then by 4 weeks, you see at the root as well as some young leaves. It's not till week 6 you get the whole plants infected. So what this tells you is that, it's really difficult to see where your plant above the root might look healthy, but it's really important to test, but where you test it is also really critical. So the root is the first instant to test if you suspect that you have this particular virus, right? And then you're not going to see any symptom until you get week 6, and by then it's too late. Yeah. And so it's important, just like the other 2 diseases that we mentioned, keep your system clean, disinfect, and also, right, when you get cuttings, seeds, and whatnot, make sure you get those tested, get it from a reputable source so that you're not going to get contaminated. Luckily, this virus, even if you have it on the flower, has no effect on us, a human. It's harmless for people.
We're at this stage of the industry and where we're at with the maturity of propagation, your nursery that you're procuring clones from should be able to provide pathogen screening results. They should be confident in what they're telling you about their pathogen screening regimen. If you're working with a TC lab, they should be able to provide you with that triple viroid assay result for that mother stock plant that you're about to bring in. So, great point. Thank you for bringing that up. And I'm actually curious, roots, leaves, what about homogenizing a root and leaf sample if you are that concerned when when going through that?
Yeah, for sure. Definitely I would do sample both. Like as you said, Sir Joe, sample the root as well as the leaf. Because you don't know where that, but usually it will hit the root first, but I would sample both. And frequency, it's also important. I don't have that slide here, but Dr. Panja, what he did was that he actually showed when he frequent sample testing, it can actually reduce the virus from, I believe it was from 35% down to 7% over a number of weeks. So the sampling, because once you sample, that plant or particular plants are affected, you can isolate them, right? Remove them, right?
Right, and that's a great point, right? Testing it one time, then you wait 2 months to test it again. Like, what's the lifespan of a mother? It should be around 3 to 4 months. Then you repopulate with the fresh, healthy Gen 0 or an in-house source. So that's a great point. Thank you for bringing that up. Is the frequency of testing, because this is a latent viroid.
Yeah. Exactly.
Yes.
Right. It's going to express whether you're just going to wait it out or you're going to put it through stress, environmental, pest, nutrient, whatever it might be that could trigger that viroid from actually expressing and really causing some problems.
Yeah, and as the name said, right, it came from the hop industry, and this virus has been around for, probably since the 80s or even before then.
Yeah.
And if you're growing from seed as well, that does not make you safe. It can actually live in seeds for years as well. So if you're getting seeds, make sure those are from a trusted source as well, and always test those too, because it can really affect everything.
Couldn't agree more. Like, I've been hearing rates up to 35% transmission rate through seed progeny.
I've heard up to 50.
So, guys, this is also the level of the maturity of breeding, right? So if your breeding projects are not starting with healthy material to start those projects with, you should reconsider how you're starting those, right? Make sure you're testing your parents. Make sure you're testing those before you make seeds. Be responsible with it. Like, we have the tools. Yeah. It's 10, 15, 20 bucks for a hop latent viroid sample. Yeah. Come on, be responsible with it. So.
And back up those genetics too. If you have something that's fire and you want to preserve it and you have the ability to back it up, back it up, because it takes time, and by the time you find out that you have latent in that strain, it's going to be too late and you're not going to have it to run. So back it up before you have a problem.
And I don't want to divert too far from the conversation, but meristematic initiation versus nodal initiation, there's a big difference here. So make sure that when you are looking at your tissue culture lab partner, you do ask those questions. Is this meristematically initiated, where you're completely dissecting the meristem off the vascular system to make sure that you're able to clean up all the disease from it? Or is it a nodal initiation, which you're going to get it a lot faster, you're going to get it much more rapid, right? Obviously, that's where you're looking at the potential risk of passing along more disease. So.
Okay, so we've talked a lot about the problems and some tips and tricks for how to potentially avoid them in your facilities. So, of course, be careful of overwatering. Hard to do if you're growing in DWC, but if you're not, then that's always a great option. And also to ensure proper drainage while you're growing. And that kind of leads into using high quality mediums. So if you are using a soil or a peat or a cocoa, using a high quality medium can help prevent you from bringing in pathogens and pests with the medium itself. I've seen a lot of that with some of the cocoa that's brought in internationally. So keep an eye on that as well as your temps and your humidity. And like we said, high humidity and high temperatures can help the pathogens spread faster and progress a lot quicker, so keep an eye on those. And automation is great, and a lot of people have engineering controls set up in their facility to monitor these things, but it's always great to have some kind of fail-safe, because I've heard that the power goes out often in California, and automation, electronics, they're fantastic, but they're not foolproof. As the lights just go out. It's not foolproof, right? So again, things like high shield, things like our high shield product, which we'll get into a little bit later on, can also really help to prevent environmental stress when your automation fails or when your engineering controls fail. So that's really important as well. And last but not least, proper sanitation protocols and just your SOPs and your protocols in general. Doug touched on it a little bit earlier on when we were talking about the diseases, but if you do have an infected plant, isolate it as soon as you can, get it tested, and then if you're keeping that plant in, make sure that's the last thing that you're seeing in a day. Like, don't be, going back and forth in your rooms, and just be really mindful of where you're moving in your facility and what you might be touching and spreading to your facility or someone else's as well. Yeah. Does anyone have any questions about thus far before we move on to products a little bit? Gonna take
I think we're good.
Nobody? We're good? Okay, so before we dive into the product a little bit that you've seen up here, kind of give you one slide, about kind of product development formation. I'm a chemist by training, so I have to give you a little bit about when I go look at make product, what thought we put in it. So like just a general process. So product generally contains an active ingredients plus inerts, right? There are different inert ingredients, and then together, you put those two together, hopefully you get a commercial product. So the idea is when you get a good product formulation, it's important to get a product that is safe, effective, right? And easy to use. Those are really, really critical, right? And the active ingredient are the ingredient that does the business in your product, right? And so you have to look at the property of the active ingredient. Is it going to be water soluble? So if it is, then I'm going to use a water-based type of chassis to dissolve that active ingredient. But if it's not water soluble, it might be oil soluble, or it might be some other phenomenon, right? So we have to look at the property, physical chemical property of the active ingredient. The inert ingredient is also very critical. The inert ingredient helps the active ingredients to perform, right? So if you have a product that's a folio spray, if you don't have the proper spreader or sticker, when you spray, it ain't going to stick on a leaf. It's just going to fall right off. Yeah. So it's going to be useless, right? So an analogy I always like to look at is like, say, aspirin, right? The active ingredient in aspirin is acetylsalicylic acid. Well, you can't just take that and then expect it to work. Aspirin has like 5% of that in there. The rest of that stuff is what they call excipient in the drug development world. And those actually help to make the active ingredient work. So when you see a product have the same active ingredient, let's say 2 different products have the same active ingredient, doesn't mean they're going to work the same. If the inner ingredient makes it more effective. So we always try to do that to make it more effective. If they're using as a foliar application or a drench application, that's really, really critical. So the first product that we're going to talk about is Hygrozyme. So obviously Hygrozyme has been in the market, as Leanne said, for almost close to 20 years, right? So Hygrozyme really, it's a root cleanser that really helps to prevent root rot, right? So it's a preventative measure, right? And by doing that, you're going to help to keep your root nice and healthy and clean. And this is a picture, I believe, from one of our grow that uses a DW.
It is, yeah. DWC grow.
DWC product, right? And so it makes your root right off the bat nice and healthy, right? And obviously, by doing that, you're going to get nice and a lot of good yields. And we have all the enzymes, the critical enzymes listed on our product label that's shown there. So the way that enzyme work is essentially by what we always like to say, a lock and key theory, right? So the enzyme has specific active sites or shapes where the substrate that fits directly in there. So in our particular case, the main enzyme here is cellulase. So cellulase is very specific for probably the second most abundant polymer on the earth, cellulose. So it clips cellulose into specific simple sugars. And those sugars, in essence, also acts as a nutrient feeds for your plants as well, too. And in the next slide, before Leanne goes to the next slide is a little animation. It's what we call a tissue test. So there are 4 products. One of them is Hygrozyme. The other are also enzyme product on the market. And we call this the tissue test because the tissues are mostly composed of cellulose. And you can see the different type of product, how they actually degrade the cellulose with their enzymes.
Yes.
What do tissue and dead root matter have in common? They both consist of cellulose, a compound that can only be broken down with one key enzyme, cellulase. When choosing an enzymatic product for your plants, you want to be sure that it has the specific enzymes to target and break down the cellulose in the dead roots. The more units of cellulase in an enzymatic formula, the quicker it will help to break down the dead root matter, preventing root rot and increasing nutrient uptake in the plant. The tissue test is the easiest way to determine the effectiveness of different enzyme formulas in the market, ensuring they contain the correct enzymes to clean and accelerate your root zone. The most effective enzyme formula will break the tissue down into small fragments and will do so quickly. Enzyme formulas that do not break down the tissue at all do not contain cellulase and will not break down the dead root matter. To truly help your plant's health, make sure you're investing in enzymatic formulas with high amounts of cellulase listed on the label like Hygrozyme, North America's #1 horticultural enzyme formula. To learn more about Hygrozyme's other benefits, watch our video, How to Accelerate Your Root Zone with Hygrozyme.
So you can see it completely broke down.
Exactly.
Yeah, completely broke down the.
Yeah. Perfect.
And if you're interested, to try the test yourself, reach out to us. We'd be happy to send you the tissue test kits.
Yeah, 100%. So the next product that we're going to jump into is actually our newest product. So this just launched into the US this past June, and this is Hygroben, which is our beneficial microbe, and we're really, really excited about it. So it is a beneficial microbe with 5 species of Bacillus bacteria, and it's formulated to increase yield, increase your terpene content, and increase your cannabinoid content as well as just improving and enriching the overall environment that you're growing in, especially if you're growing in a sterile environment that doesn't already have microbes and beneficials in it, like growing in the ground and soil would. This is a really great option to get them in there and add them in. And it also works really synergistically with Hygrozyme because Hygrozyme will actually feed the microbes in Hygroben as well. So they work really, really well together. And if you're not familiar with what Bacillus is, it is a rod-shaped bacteria that's mostly aerobic, which means it really loves oxygen, and it thrives at 25 to 30 degrees Celsius, which I just realized I'm in California. But I believe that's the low 70s, if I'm not mistaken. And it's naturally found in soil, water, and air as well. So what Doug has done and his team is isolate these specific species that are found naturally and then use them in this product. So again, with our label there, all of our products have this, but all of the active ingredients and their activity levels are listed directly on the label, so you know exactly what you're getting and what's in there. And then those are the specific 5 species that are in there. So we have Bacillus licheniformis, Bacillus subtilis, Bacillus pumilus, Bacillus amyloliquefaciens, and Bacillus megaterium as well.
So a quick question about that. We've heard the term synganic be thrown around pretty recently, right? Hydroponic growing in sterile media, and then introducing microbes. Is this a good product that you can introduce to try to be in that synganic approach?
Absolutely, absolutely. It works really well in a synganic approach, and that's what a lot of our clients and a lot of our customers are doing when they're growing. We do have a lot of people that are growing DWC, and then they'll add in things like Hygrozyme and Hygroben together to really create a really strong, healthy environment.
Any questions? One back here. Thank you, sir. Name and where you're from.
Hello, I'm Mike. I'm from SF. Just a quick question. Comparing to Hygroben versus like the Impello product like Tribus or like the Emerald Goddess Root Wizard, they both contain around the same microbes. Can you go a little about like what differentiates Hygroben versus like the competitor itself?
Well, as I alluded in my slide on formulation, right? So yes, some of these Bacillus species are in other products as well too, but really it's also the inert ingredient that's helping, right? So obviously I can't disclose the formulation for you guys, but it's really the inert ingredient that makes the active works really well. And then as Leanne will show you some actual data that trials that we done with a third-party independent contractor, and then you can see the result from there, and we can talk a little bit more about it as well after.
Awesome. So how exactly Hygroben works is through 3 different direct mechanisms, which are nitrogen fixation, phosphate solubilization, and cytorophore production. And basically, through the helpful teaching of Doug, I have learned that these are basically just fancy scientific ways for saying that the bacteria are going in and they're converting these materials, whether it be nitrogen or the phosphates or cytorophores refer to the iron, and they convert those materials that are present in the soil or in the environment into forms that are more compatible and bioavailable for your plants to use. So basically, it's just going to make your nutrients and your fertilizers more soluble and give it better uptake.
Yep. Got one question back here. One sec.
Yeah.
How you doing? My name is Alex. And how does high oxygenated water, like if you use like a Moliere nano bubbler, does it affect your microbes or anything like that? Like somewhere like in the 25 parts per million up to 40 parts per million.
It should still work in those systems. Like the microbes should be still alive in the systems. And so we in terms of the usage and the application intervals on our label, we apply that pretty small volumes, like 1 mil per liter, and the application interval is once a week.
Next question is there a certain threshold? A lot of people use like products like hypochlorous acid. Is there a certain threshold that your bacteria or stuff like dies or lives, thrives?
Very good question. So we actually have tested that, and we have tested in hypochlorous acid. I won't name the products, but we have tested, and then we have tested according to the product's low label as well as its high label, and there is no issues in terms of the bacteria having the total, meaning their CFU count doesn't decrease after we subject that to those hypochlorous acid products.
Is there any video or with a microscopic video that would show like your bacteria or your enzyme living in water that has zero hypochlorous acid in different stages or percentages of hypochlorous acid until it affects it? You know.
We don't have videos, but what we did was time where we have basically taken the product, incubated with different concentration of hypochlorous acid. One of the product on the market that has hypochlorous acid as the active ingredient. So we did it at the lower dose of that hypochlorous acid product and a higher dose. And then because we know the total CFU count of our product, then after we subject it to that over a certain number of, say, days and whatnot, we then have the sample analyzed, and that count, the CFU count, which is the unit to measure bacteria, does not decrease.
Okay, thank you.
Great questions. Okay, so next up, like Doug said, we did some testing on Hygrozyme and Hygroben being used together prior to launching this product. So we worked with some third-party laboratories, and we did some blind testing on the use of a control versus Hygrozyme on its own, and then a control versus Hygrozyme and Hygroben on a variety of different factors. And what we found was that by using the two products together, you can increase your dry flower yield by up to 30%, your total cannabinoids by up to 20%, CBD on its own up to 20%. This was on a CBD dominant strain, as well as total terpenes up to 20%, and then beta-myrcene and alpha-pinene respectively 35 and 35% increases. Yeah, so of course, we all know this market is very competitive. It's a really hard time to be a grower, and it's not an easy industry to be in, and everyone's looking for an advantage. So these kinds of products and anything that can get you a higher yield and increase those terpenes and increase those cannabinoids and get you a higher quality product is going to give that advantage that you really need. So next up, we're going to go into our High Clean product. This is my personal favorite product of ours. It's a natural irrigation line cleaner that does not contain any hypochlorous acid or bleaches. So it's completely safe and compatible with all beneficials, as well as it will really help to reduce time and labor for your deep cleans in between cycles. It prevents clogging and buildups in your lines from nutrient and salt buildups, as well as it can increase the overall quality of your finished product because it's really going to flush through the media and give you just a really nice, clean, solid product at the end. And basically how it works is the product goes in and it's going to bind with the ions from excess nutrients and salts and basically make those more soluble so that they can be pushed through the lines a lot easier. Yeah, anything else to add, Doug?
No, that's it.
Okay. So I do have a little bit of a video here showing kind of how the product works. And so what we've done is in each of the little vials with the caps on them, we have simulated hard water, not because you would necessarily be using hard water in your facilities, but more so to simulate that buildup that might be in your lines from using salt-based products or nutrients and so on and so forth. And then we've added high clean to the vial in the middle from the beaker in the middle, and then we have 2 common competitor products out there as well. And yeah, we will see how it works to clean all of that up. So yeah, you can see from the video that the one in the middle that has the high clean is much clearer than the one on either side of it. And that's because, again, that high clean product is just going in and making all of those nutrients or the salts and calciums that are in that simulated hard water more soluble so that they can just dissolve and be passed through. And again, like Doug said with the tissue test, we do have kits available for this. So if you want to see it firsthand, we'd be more than happy to send you one. Just reach out to us and we can send one over.
Out of curiosity, what ppm did it break it down to after you did that test?
The ppm it started at was 2,000, and then what does it break it down to, Doug?
It's probably like below, probably around, like I think around a few hundreds, like around there if I remember correctly.
Could you repeat what was on the water? What was in the water originally, and the milky water?
Sorry, can you say that again?
It's hard water.
Yeah, it's simulated hard water. So this is like there is a recipe out there actually on the EPA's website that you can actually make hard water through different chemicals. So you simulate hard water, and then as Leanne said, the ppm of that hard water is somewhere between 2,000 and 3,000 ppm.
So you're saying that like if you use high clean, it would help not only clean the drip lines, but also the membrane inside, like the drip emitters, like 0.3s, 0.5s. So
Oh, yeah.
Clean it up and
Yeah.
Increase flow again. Okay.
Exactly.
Yeah, exactly. Yeah.
And you can do that during your run. You don't have to wait till after the run.
Precisely,
Yes.
yeah.
Yeah. So on our label, we have usage for a while during the run, but of course we also have usage after, in between grow. So often your line is very dirty after. So if you want to get your lines nice and clean, you use basically use a larger like a larger dose to clean it, and that's all in the label, and then that will get your lines nice and clean.
How often was the question?
So during the grow, you can use it right from the beginning until the end, right? And then for cleaning your line, I guess it depends. Like some people clean their line after every grow, but I know some don't. So I actually had a guy up in Vancouver, not necessarily on cannabis, but he was running a nursery using a drip, using a line. He cleans his line once a year. Like, I mean, the line is just like...
We don't recommend that.
The line is just so dirty. So he used some of this stuff, and he just left it in there. So we do recommend, like, obviously it depends how dirty your line is, right? So the best is to actually leave it in there, let it sit in the line for 24 hours, And then after that, you flush it through, you rinse it with water, and then you'll be good to go. So, and you can, right now on the label, it's around 2 mils per liter for in between grow. But during a grow, it's much less. During a grow, we only have it as 1 mil per gallon.
Yeah.
So it's very, very low.
That's very concentrated then. That's incredible.
Yeah.
Yeah.
So, I mean, that definitely raises some curiosity. Just because there's a lot of pathogens that can build up during the run. Again, there's a lot of challenges that happen during the run.
Yes.
Environmentally, whatever it might be. So.
And the nice thing, right, as Leanna already mentioned, that it's compatible with beneficial. You know, a lot of people use it beneficial. We test it against trichoderma, bacillus, mycorrhizae. So there's no issues in terms of compatibility. So it doesn't affect you beneficial products.
I'm gonna ask the crowd, what's a common drip line flush that you would use? Because I heard Zerotol is a good drip line maintenance flush. What else? Pretty common. Bleach, right? Hypochlorous acid. Like, how does this compare to some of those products?
So those are like those will definitely affect your beneficials, right? As I'm sure some of you may not know, right? And I would put high clean head to head against them in terms of removing the buildup of salt and mineral, and also help clean your emitters. Those will not do that, and high clean will definitely remove those excess salt and mineral of your line.
Not just that too, but a lot of the harsher chemicals can also really affect your terpene profile, as well as like the outcome of your ash at the end. By using High Clean, because there are no harsh chemicals in it, you're not going to affect the terpene profile whatsoever. Your flavor is going to stay intact, and we also have people reporting really, really white ash when using this product as well. So really, really clean end product.
Wait, you're saying white ash impacts the flavor of the smoke?
And the last product I'm going to talk about is High Shield. So what is High Shield? Well, High Shield is really it's a plant immune boosters made from chitosan, which is derived from the exoskeleton of things like shrimp, crabs, and lobsters. So really, it's environmental friendly, right? And some of the benefit of High Shield, as Leanne might have alluded to earlier, it helps it reduces what we call abiotic stress, like heat, cold, things like that, right? And helps to also reduce transpirations, and it really just kind of boosts your overall plant health. And the way that it works is that it actually triggers the plant's SAR, or plant systemic acquired resistant. I like to think of it as like it actually helps the plants to prepare or tricks the plants that has been attacked by pests or pathogen. So it's get them prepared to be alert, essentially, for an attack, right? That's one of the nice thing about this product, and it's all natural. And
I'm just going to jump in there really quick. If I could use one analogy, I'd probably compare it to like a vaccine, very similar to like a flu shot. If you go and you get a flu shot, it doesn't guarantee that you're not going to get the flu, but it will kind of help with reducing the symptoms and reducing the length of the systems. And that's very similar to how High Shield works as well.
And another benefit of High Shield is that, so High Shield, as I said, contains chitosan. Chitosan is probably the second most abundant polymer, but the particular chitosan we use is actually high molecular weight. So what that does is actually has a physical phenomena as well too. So it actually forms a little, I don't know if you can see in those microscope slides there. So those are basically after one application of the product, and after two application, and the water evaporate and look at it under a microscope, you can see those little specks. So those little specks are little films on there. So imagine you spray this, and we do recommend you spray it during the flower stage. And when you spray this on the leaf, you imagine that on the leaf. So what that does is actually kind of alter the leaves properly a little bit. So if you have pathogens or spore flying around, it's going to have a harder time adhere to the leaf as well too. And above the, which I didn't mention, we have some people use it as a spray. You can see another phenomenon that people are seeing is that you actually get a much thicker stalk, right, compared to untreated. And that's a foliar application. Of course, by having that, you can handle a larger, heavier canopy.
Question, is there a particular micron that you should be spraying versus fogging?
It's generally we find spray works much better, both because of giving that physical full barrier. So there are some chitosan product out there that they use low molecular weight chitosan. So when you use those, you don't see this physical phenomena. And there are actually chitosan product out there. Ours is not for that. There are actually chitosan product out there for post-harvest preservative food grade. Like people spray it on your traditional crops, and it actually helps to maintain the shelf life of whatever food last longer versus an untreated.
Okay. And that's all we have for you guys. What I will say as well is Doug and my contact information is up there. I know I have business cards with me. I believe Doug does as well. And we do offer commercial trials to anyone that's interested. So if that does speak to you, if any of the products that we talked about tonight intrigue you and you'd like to give them a try in your own facilities, please do reach out to me. I coordinate all of the trials for Hygrozyme, and I'd love to get it into some of your hands. And in addition to that, we do have some boxes, sample boxes just around the corner here with a 500 milliliter of each of our products for you guys all to take home, as well as a hat and a shirt. So you guys can give them a little bit of a try. But like I said, for bigger samples, please reach out to me and I'd be happy to help. Thank you guys so much for listening. And if anyone else has any questions, the floor is open.
Yeah.
All right, what questions do we have? We're right back here.
The shelf life for Hyclean?
2 years.
The shelf life for Hyclean is 2 years. Yeah.
Next question.
Mixing order for Hyclean? Like if we're in the dosatron line itself, where would you recommend placing that Hyclean at?
That's a great question.
That's a good question. So if you're mixing using a dosing system, I recommend you have it as a separate tank, right? So you want to have the final concentration of Hyclean going into your plant at 1 ml per gallon. Yeah, that's a really good question, and I had a few of those talking to grower in person about that, because everyone's like, some people may not have enough tank for the dosatron, but the best is you can dedicate a separate tank for the dosatron.
In general, if you're not using a dosatron, then you would add Hyclean last. Is that right, Doug?
Yes.
Yeah.
Good question, thank you. What else? Right up front.
How long has Hygrozyme been on the market, and have you guys changed the formula over the years?
Yeah, great question. So Hygrozyme has been out for almost exactly 20 years at this point. And I mean, as far as I know, we have not changed the formulation in all of that time. It's still exactly the same. So
Exactly the same as, about 20 years ago.
Any questions back here? Let's hear it for Doug and Leanne. Thank you, guys. That was a lot of information, very good information. Obviously, there's 4 products here. They each apply towards something specific, but again, it's all about using it in your run, post-run.
Yeah.
Great stuff here, guys. Again, thank you all for coming out. Really appreciate you spending time with us. Shout out to Hygrozyme and the crew for flying all the way out from Canada. Really appreciate you guys making the trip out here to come spend some time in Sacramento. Thank you. Let's have a shout out for our booth sponsors that were here tonight with VitaWool. We had Grodan, we had Friendo, we had Kayak Horror, and of course, Indoor Sun Hydro. Thank you guys so much for having us. We'll see you next month. Much love.